Hello everybody, my name is Erin Neff and I am a senior neuroscience and psychology major, and I would like to introduce my partner.
Hi, my name is Ellen Kosajeski. I am a senior neuroscience and psychology major.
In creating the hypothesis for our study, Ellen and I were interested in how pediatric concussions affect emotion. However, in our research, we found that the standardized image database that Ellen will be talking about in a minute was normed on valence and arousal in adults, not children. Additionally, literature has shown that children may lack the worldly knowledge to fully evaluate an image like a swastika. If you have the historical background on what a swastika means, in an adult, that may elicit angry or negative emotions. However, in a child, they may not fully understand what that means. We also were finding studies where parents were removing their child from the study due to the presentation of images like gore and death. Since we eventually wished to study a clinical population where sample sizes tend to be smaller, we wanted to mitigate the risk of participants being removed from the study. Thus, we developed Study 1 to address these cited problems, using animal images that do not require the worldly knowledge that a swastika may bring.
The aim of our first study was to establish a parental-approved, developmentally appropriate set of images for children from the ages of 7 to 17. This dataset was then going to be used in Study 2. Parent and child pairs completed this portion of the study. The pairs consisted of any one parent or guardian and 1 or 2 children. We utilized 221 images from the NAPS database and the 9-point SAMS mannequin scale to rate the images on valence and arousal. Valence refers to the positive or negative feeling that the image incites, and arousal pertains to how calm or excited the image makes the person feel. The valence scale varied from very unhappy to very happy, and the arousal scale went from very calm to very excited. The fish image below is an example of the animal image that they would have seen. Below it is the scales that the children used to rate each image. First, everyone consented or assented accordingly. Then the parents would answer yes or no to the question, would you allow your child to view this image? For 220 of the animal pictures. The parents were instructed to keep in mind that this would have been for research purposes. If they had 2 children completing the study, they would rate the image for both children at the same time. The first child would then rate 100 of the approved images. If 2 children were participating, the second child completed the portion after the first child did all of their ratings. Then they were debriefed. We applied exclusion criteria to the data. Each image had to have at least 8 ratings from the kids, and 96% of parents had to agree to letting their child view this image. Out of the 220 images, 80 total images were retained. None of these images had a negative valence rating. Therefore, we utilized the 30 lowest rated neutral images and the 30 highest rated positive images for the next study. The graph shows the average ratings for neutral images and the average ratings for positive images of the 60 images that were used in Study 2. Study 2 utilizes electroencephalography, EEG, to measure electrical brain activity. The brain activity measured at a specific stimulus or event is considered an ERP, event-related potential. Research has discovered that certain activities designed to test the same cognitive abilities elicit the same ERP. The late positive potential, LPP, is an ERP marker that indicates a person is paying attention to an emotional stimulus. Study 2 used EEG on a population of neurotypical children to determine if our stimulus set from Study 1, the 60 animal images, can evoke an LPP response.
Using a 128 electrode EEG net, our pilot study consisted of 5 children between the ages of 7 and 17. Using the 60 images from Study 1 with a distinct valence between neutral and positive, we instructed children to passively view 2 blocks with the same 60 images repeated in each block. The times are outlined in the procedural flow diagram below the method section of Study 2. Looking at the 4 LPP ERP plots under the results section, we are able to see an LPP elicited at the frontal electrodes, FC, parietal, PZ, and occipital electrode sites, peaking at 200 milliseconds after the presentation of the image, and is sustained for approximately 1,000 to 1,200 milliseconds. As Ellen mentioned before, the LPP is a marker to attention to emotion. We hypothesized that there would be a difference between the amplitude of the LPP between the positive and neutral conditions. However, due to our small sample size, we did not find this effect. Because the results were preliminary, future studies will expand the population size to establish the LPP differences between neutral and positive conditions, as well as begin testing children who have a history of concussion. If you have any questions about this study, please feel free to email either myself or Ellen. Our emails are at the bottom of our poster. Thank you.